So today's topic is the eukaryotic gene prediction. And then we would like to start from scratch, meaning that from the sequence, without giving any experimental data in it. Experimental data could be there, but in the way of doing the training. But for my sequence that I want to investigate right now, I don't want to give any experimental evidence for that. So basically, for eukaryotic gene prediction, I just want to give a sequence and then get the result. So since I did not give any hint or experimental evidence, I can say that I started from the start. So we say, in this case, the ab initio or de novo gene prediction. So everybody understand the concept here? The complexity for eukaryotes compared to the prokaryotes are eukaryotic genome has multiple exons. Exon 1, exon 2, exon 3, like that, right? But the prokaryotes usually all the way without the multiple exons. And the alternative splicing. Is there anyone who can explain what is the alternative splicing? Yep.
Alternative splicing is a mechanism through which one gene sequence can give rise to different variants of mRNA. That is because the pattern in which exons bind to each other determine what the final gene product will be.
Yeah. So even though the DNA sequence is the same, due to the alternative splicing, actual like an mRNA sequence could be different. So it's complicated compared to the prokaryotes, which doesn't have the alternative splicing event. And then 3' UTR untranslated region, and then 5' UTR where the promoter binds, and those are very complicated compared to the prokaryotes. In the prokaryotes, there are relatively simple motif. And then those sigma factors are pretty well known, and it's relatively simple. But in the eukaryote case, how the gene expression is started, it can have a multiple promoter, like the promoters, and then RNA polymerase 2 is recruited, and then it makes some curvature there, and it's somewhat complicated, much more complicated. So due to this complexity, if you use a glimmer which did not account for all this, your prediction is going to be wrong, even though you get some prediction. So that stress out that we need the eukaryotic specific gene prediction software. So that's what we are going to talk about today. Before we move on, I would like to give us some the content sensor and signal sensor. Let me start with the signal sensor. So eukaryotic genome has some specific, the signal in the sequence. Everybody heard about the poly A tail, right? Of mRNA, right? Yeah, it's a definitely it's it gives a signal, some signal or some information, and it's a like that. And then everybody know that after that the gene is the mRNA is done, right? So those are the typical signal sensors, and promoter and all these are the signal sensors, examples of a signal sensors. How about the content sensor? It relates to some features. inherent to the coding sequence base composition. For example, everybody probably know that 3 bases code for 1 amino acid, right? And let me ask you this question, the third one, that two DNA bases followed by the third one. There is a third one. Is third one completely random, or is there any like bias for the third base? If the region is somewhat intergenic region, yeah, it looks more like a more random. But if it's in a coding region, it is not. Yeah, it's not totally random for the coding region for the third base. But if you just look at the some other regions other than the coding region, it could be somewhat more random. So what about the not the trimer but hexamer? Hexamer means a 6 nucleotide. A is an example of a hexamer. ACT is another example of a hexamers, right? So the frequency of all possible hexamers, are they random or not? Yeah, it depends on the region. You are talking about the content sensor. It depends on, it's not totally random. So those software can censor those and then utilize that in doing the gene prediction. So that's why I introduced the content sensor and signal sensor concept. But in reality, you don't need to worry because those concepts are incorporated into the software. So you just apply the input file, and then you get the gene prediction result. But some software heavily use the signal sensor concept more. Some software use the content sensor to do the gene prediction. Okay. And comparative genomics is actually, you can compare genomic sequence content between different species. For example, human versus mouse, or you can compare human versus C. elegans, the round one. And by doing this comparison, you can find out conserved region, or you can find out variable element, and you can learn about how the gene expression is actually controlled by doing the comparison. So the take-home message for the comparative genomics is, by comparing genomes of a different species, we can learn certain things. So that's the purpose of the comparative, especially about the function. And you might wonder why I introduce this kind of some comparative genomics in this eukaryotic gene prediction. Human genomics is pretty complicated. By doing comparison to more simpler organism like yeast, you can actually learn something from there. The yeast genome cannot explain 100% of the human genome, but certain important features can be actually explained. So when you have a very complicated the organism like a human, we usually start with a simpler model organism like a yeast or C. elegans, and then by doing the sequence comparison to those, and actually for the human, it's not easy to do any biological experiment mutation, but it's pretty easy to do the mutation experiment for the yeast or C. elegans. So we know a lot more in detail, not on the human, but on the this model organism. But by doing this comparative genomics, we actually learn a lot about eukaryotic genome structure, especially the complicated human. Now, I would like to do some class exercise. So far we talked about the gene prediction, eukaryotic gene prediction. Why don't we do actually eukaryotic gene prediction? I will give you about 5 to 10 minutes. So this is what I want. I want you to struggle to get the sequence of DNA sequence of BRCA1 gene for human. So get the BRCA1 DNA sequence. And once you do that, can you use eukaryotic gene prediction software, what we call in this case, GenScan, and actually predict the which introns in that sequence. So now the time is 6:25 or 6:26. Why don't I actually give the 5 minutes so that you can actually work on, and feel free to work with your colleague if you want. Any collaborations are encouraged here. And after 5 minutes, I would like to ask one of you to share your screen and then show what you have done or get into trouble, and then we can discuss together, and then we will actually do this exercise together. So why don't I give you 5 minutes so you can actually try. Get the DNA sequence of BRCA1 and then try to predict introns in that, or in other words, exons. Are you done? Yeah, question? Um, yeah, actually, in the gender, there is some full, but it's not easy to find. Okay, so you want to, don't use the gender. Don't use them. If you have trouble with finding the, so try to use some other. Yeah, I would probably use the UCSC. I would show that in that. Yeah, question? Oh, yeah. Thanks.
Recording in progress.
Yeah, question? Oh. I don't know how to use GCSs. Okay, let's do the quick, can you, how many of you actually finished the step one? Yeah, how many of you tried the step two? Probably the last name. Okay, don't worry. Let's do it. Yeah. Is there anyone who wanna? Yeah.
But business says that there are two pieces. I think that's
Yeah. Let's see. Let's see. All right. And do you want to share your how you have done it? Okay. Let's try that. Let's see. So the first step is to get the breakout sequence.
I already downloaded the sequence. So
Yeah. How did you get that? The breakout sequence. How did you get the breakout? Mhm.
and then I can search for this.
Partial closing sequence.
Yeah. Maybe that's the
Yeah, it should be the much longer than that.
So the first
No, actually, right. Yeah. Why don't I actually share the way how I done it? Okay. What's your name? Megan. Megan tried to find the breakout sequence from NCBI database. Actually, that's possible, but the NCBI database somehow contains a lot of partial sequence too, in addition to the full sequence. So it's fine, but you have to do some like a digging to actually find the full length. So in bioinformatics, there are so many ways to do that, and then results are slightly different depending on which method you are doing. But overall, it's about the same tendency, but it's not 100% same. But let me show you one way that I'm doing if I'm asked to do find out the breakout sequence of a human. UCSC genome browser. Can you yeah, let me share that screen. So let me first focus on the step 1, getting the BRCA1 DNA sequence. UCSC is the University of California, Santa Cruz, the genome browser. Anyway, I'm going to introduce this in detail later, not this week, but later in the course, some more detail. But can you Google this and then come to this website? Let me share this website. Right? Yeah. I would like to try the genome browser. So there is a link called the genome browser. Do you see this link? So I'm going to click it. And yeah, please do it with me together because it actually helps. And making sure that you are studying human, right? I click the human. And then when I click the human, there are multiple like genome. And then the default one, the most popular nowadays is this HG38, human genome version 38. So let me just keep that most popular version. And the position or gene name is BRCA1. And when I type the BRCA1, it has two like entries. One is BRCA1, and another one is BRCA1's pseudogene, not real like expressed part, but similar sequence is located in somewhere else, and it's not what I'm looking for. I'm looking for the real BRCA1. So I'm going to click not the pseudogene, BRCA1 P1, but I'm going to choose the BRCA1. Is it clear? So now I came here. Everybody on the same page? I want to view DNA sequence. Do you see that the view menu is up here? I want to view the second one, the DNA. Okay. And then I want to use the default setting, and I want to get the DNA. So I'm going to click the get DNA. Yeah, Google somehow thought this is a Somali language, but that's okay. So this is a typical FASTA file, FASTA format. Start with the greater than sign, followed by unique ID, and then now the sequence is there. So I'm going to Control A, Command A to copy, and Command C to what is that? Command A to select all, and then Command C or Control C to copy. And then if you look at that, let's check out how long is this calculator. It is located in the chromosome 17 from this position to this position. So I want to subtract these two numbers. So this one minus this one equal this one. And I have to add one base. Why? Because you want to include both ends. So that's why I have to add one more plus one. So BRCA1 gene, according to this UCSC genome browser, HG38, it's 81,070 bases. So when the Megan said chose the that 3,000, 4,000, that's way too small. Yeah, because it's a really small partial sequence. And if you choose that small sequence, you cannot have that like intronic structures in it because it's a very small part of it. So now we have about 81,000 base pair long, right? Any questions so far getting the BRCA1 sequence? Everybody got it? Good. So when you use the NCBI, just be careful. If you are not careful, you end up with getting the small partial sequences. And then in the beginning, you might think, yeah, why don't they just put the long standard sequence like a UCSC? Why did they put the partial sequences? Why? This sequence, BRCA1 sequence is reference sequence, someone's sequence, right? But your BRCA1 and your BRCA1 is not 100% same as this, right? So by having a lot of partial sequences, you can actually have some sequence like a variability. And yeah, so personally, I don't mind having a lot of partial sequences and other like origins of a BRCA1. But when I actually have to do some like gene finding, yeah, I want to use some standard, HG38 standard. So that's how I got it. Now, let me move on to the second step, that run the gen scan. So here is a gen scan website. I copied and I just paste it. And it should work, but I want to delete the first line. I just want to have all the sequences. Right? Now I would like to run the gen scan. Now I have poly A tail and then terminal part and intronic part like that. And then even translated protein sequence prediction. Okay. And I have one question. Why the poly A tail comes first in this case? Because if you look at the BRCA1 gene, do you see this directionality? It's not located on the plus direction. It's located in the minus direction. So actually, poly A tail is located here, which has the the shortest, the smallest value in the coordinate system. That's why poly A tail comes first in that gen scan. Yeah. Question?
Does gen scan also annotate exons?
Basically, when you annotate the introns, yeah.
Yeah, but like
yeah.
Because like if you go back to like when you input it into
Yep.
gen scan, it says no exons now.
Yeah, that's so let me show this just one moment. I want to show the further reading part. the GenScan actually output the p-value, and unfortunately, the p-value that we got is not that high, I guess. Some is high, but not all of them. And unfortunately, no particular like a really reliable exonic prediction cannot be obtained for this sequence. So it predicts something, but the prediction is not like a very high probability, p-value greater than 0.99. Pardon? Why? This the GenScan's algorithm check the sequence property, but that algorithm couldn't somehow does not have a high enough like a confidence for this BRCA1 region. Yeah. And as this was written here, when the p-value is greater than 0.9, are almost always exactly correct. However, most of them are the probability does not go like that high. So p-value column is here. In this case, the very high, but everything else is pretty intermediate or like that. It's extremely low. So this actually proves how the like a de novo prediction is hard and sometimes could be wrong. So here is actually the somewhat validated the exonic structure or intronic region. So by comparing that position to that GenScan, you can actually find out how reliable the GenScan was.
Like it's like a score.
Yeah, p-value is a score. Yeah,
Like it's
yeah, they have their own statistical model. So we usually trust that p-value greater than 0.99, yeah, which is somewhat weird. I probably would choose it the other way, but somehow the GenScan authors did that way. Okay. So now, if I just wrap up what we just talked so far, we talked about the eukaryotic gene prediction, why it can be tricky. However, why don't we actually do the gens eukaryotic gene prediction starting from BRCA1 sequence? And I hope that you guys made it. And any questions before we take a 10-minute break? No? Yeah, why don't we take a 10-minute break and continue at 7 p.m. And feel free to ask me anything in the meanwhile. Actually, let me stop the recording. Yeah, for the recording purpose, let me just repeat last 30 seconds. Yeah, I didn't turn on the start the recording again. So if you can provide the expression data like mRNA information, you can make a better prediction of a gene. Um, instead of mRNA, if you can provide the cDNA information and use the spline, you can get the more accurate information about the gene structure, which one is going to be exon. So the classroom exercise that I ask you to do is here is can you get the cDNA sequence whose accession number is NM214647? Can you also get the genomic sequence of NW732498 and then run the spline so that you identify which part is basically exon in the genomic sequence. Yeah, question?
So for the genomic sequence, can we upload the file and this alignment when it drive to show it as an error?
Oh, really?
Because of the past question.
Yeah, what can you actually try to get from?
So I think probably.
Yeah. From the file, can you actually try to delete that unnecessary part and resubmit? Pardon? Yeah. That's what I actually do. So, but in the meanwhile, we try to get how to interpret. So if you succeed, please try to make it understood the results that you got. How many of you completed this exercise? Please raise your hand. Do you need some more time, or do you want me to go over? Yeah, as usual, I would like to ask for a volunteer. Can you? Is there anyone who wanna share and then show me how you actually did it? Thank you. Who is this? What's your name? Kaizen. Yeah. Thank you. Can you actually go through the steps how you have done it?
very short sequence, but the mRNA sequence is populated right now.
Yeah, this sequence, can you show me that this sequence, mRNA sequence? Introns are already gone here because it's an mRNA. Okay. So you pasted it to this prime. Okay. And how did you get the genomic sequence?
I had actually downloaded the file.
Yeah. Right. Can you click here? Yeah, this is right. Is there anyone who has a question on how to do this? And you know the easiest way to do it is just write down that accession number without even copy and paste the sequence. But the reason why I asked you to get the actual sequence copy and paste is I just wanted to show you that mRNA sequence is that all the introns are gone, therefore it's very short. But the genomic sequence is very long. So I just wanted to show you the concept. That's the only reason. In actual setting, if you know the accession number, there is no reason to not to use that accession number because just typing that accession number will do that. Right? So now let's try to make the understand this figure. The first thing that I would like to you to think about is query sequence versus a subject. Kaiden, can you highlight the query and then the subject? Yeah. Query here is the mRNA sequence. Subject is the genomic sequence. So query, in short, query is the short sequence. And the subject is like a whole genome, like that long sequence. So everybody clear about the query versus the subject. And yeah, the other statistics are like not so interesting, but yeah, can you actually click that the first one? And when you click that, where does it align to?
The start of the subject.
Yeah, but
sorry, on the back of it.
Yeah, back there. When you click the second part of mRNA, where does it align to? Exon 2, right? Then when you click the third part, it aligns to the exon 3 in this genomic sequence, and like that, 4 and 5. Do you see the opposite direction? Mm-hmm. So which one has plus and minus vector? mRNA sequence query part or subject part?
mRNA.
Okay. The genomic sequence usually, yeah, it can be either positive or negative. Positive is to make it negative, you just make a reverse complementary, right? But somehow the directionality, mRNA sequence and the genomic sequence, the direction is opposite. Why? Because gene is located in the negative direction. So can you click the segment 1 in the query sequence? This is the first part in mRNA. It actually spans first nucleotide all the way to 71, 72, 73. I don't want to count, but basically, you know how to do that, right? And that actually aligns starting from 51818 position of genomic sequence backwards to 51748 is this A. What is this position? 51747, right? You decrease. So you go all the way like that, then that is the position. Now, is this a graphical thing? Is clear, right? Transcription start site. Can we know this is just simply the mRNA sequence to the genomic sequence. Transcription start site. If that mRNA sequence is located there, yeah, it should be right aligned to. Why not? Yeah. Can you actually click the text, not the graphics here? You know the query and the subject, and then the first segment in the query is 128 base pair long, starting from query start 1 to the query finish at the 125, and the subject start at 51818 position, and then the subject finish at 51691, and then the type is it has some exonic sequence followed by the GT. Do you know what is the GT and AG? It's a typical motif of intron exon junction. So when it see the GT, hey, this is the end of that exon. And then okay. Yeah, thank you for showing the this thing. I'm sure that you guys have a good understanding on. So basically, if you are given the cDNA sequence or mRNA sequence, you can align to the genomic sequence to get exonic information. This exonic information is much more accurate than de novo prediction methods that you used in the previous session. So is everything clear? Good. Now, I would like to introduce the NCBI. Is there anyone who actually came to NCBI? Bethesda, Maryland? No? Nobody? Okay. Let me give us some brief introduction of NIH. I actually came to NIH in 2007 and did my first postdoc there, and second postdoc, first postdoc at the NCI, and second postdoc at NIDDK. And then, I was in that area for a long time. So if you come to the Bethesda, Maryland, it's worthwhile to actually visit the NIH campus. NIH is made of about 28 institutes. The largest one is the NCI, National Cancer Institute, and second largest by at this time, I guess it's a NIAID, NIAID, the Infectious Disease Institute. And NCBI is one of 28 institutes in NIH. And they are basically making databases and the PubMed and like that. And so the NCBI basically hosts this kind of bioinformatics related databases. And there are actually a lot. You use some GenBank. You used the GenBank last time, but today some of you use the nucleotide database. There is a proteins database, and there are many data. PubMed is another database that you guys often use. So NCBI has so many individual databases. And is there any good way that you search all these databases in one shot? Otherwise, you have to Google NCBI nucleotide and go there. Or you have to Google NCBI GenBank and go there.
There is a section of the search bar which is all databases, and the center of query is the result of different databases of all these.
Yep, that's right. Basically, so many people complained about this. You guys have so many databases, and is there any like a master database or master thing that you just go there and then search for everything? So let me go to this website, NCBI National Library of Medicine at nih.gov, and then search. So like, why don't you actually go there too? Shall we search for BRCA1 since we did it? So instead of going to the NCBI genes, NCBI nucleotide, NCBI GenBank, here they have what is that? Aggregates. You can search for everything, and they call these entrées. So that what I usually do is, if I have to search the NCBI databases, I first try this entrées option first, because after typing the BRCA1, if I want to check out the ClinVar or any like the DBSNP like that, or even PubMed that mentions that keyword, and this is basically the entrées. So you can do the search job in one place. Okay. And today we use the UCS is a genome browser, and I show the BRCA1 gene structure, and eventually we click the view and DNA to get the DNA sequence of a BRCA1. But that was the UCS's University of California, Santa Cruz, the website genome browser. Does NCBI has such a genome browser? Yes, they actually have. They actually have two, not just one. One is the genome data viewer. The other one is basically variation viewer, mutation viewer, basically. So they have these two. So let me show you these two, and then so the genome data viewer first. This genome data viewer at this time has over 2,350 organisms available. And then if I just show the same example, BRCA1. This is basically genome viewer, representative genome for the human, and then especially particularly HG38, the minor update P14 version, but basically it's a HG38 version, and then it's just like the UCSC, so genome browser. So it shows the representative genome and gene structures. So BRCA1, yeah. And there, NCBI has also that variation viewer. In the genome browser, you have a representative gene structure, BRCA1 here. But here, if I search the same region, BRCA1, it seems like showing the same kind of a structure, but it does have many other variant mutation related information down there. Clinical database of a SNP, and then so it shows a lot of variant information together. So NCBI has not just the Untress is basically gathering of everything. They also provide some genome viewer, like a genomic data viewer, as well as a variation viewer. And you will use this genome data viewer as well as a variation viewer in the graded homework. Do you have any question about the NCBI and then this brief introduction? Yeah, question?
Are there any folks using this over the UCSC browser or like the other one? Or is it just personal preference?
That's actually a good question. The information is not 100% the same. That's the problem. And also, the visualization that you get is not exactly 100% same. So for some aspect, UCSC has the better like the function, but in some other aspects, the NCBI has some. So you will see that so in this course, we will you will use the also, and it's not only NCBI but also the UCSC. But when you do the group project, you are welcome to stick to any one of these, or you may try to compare. It depends because the information is not exactly the same. So it's not just your preference, but certain information is better offered in some certain browser. No, actually, variation viewer, yeah. Yeah, that's a good question. I always used here for human, but does it show for other organisms? Yeah, it seems like default is human. Genome structure is the same, but the variation bureau has this section, down section, which has variations. Yep, they are actually you can actually have it there too. So it's somewhat redundant. It was not like that, but as the time goes on, it became compatible. Yeah, that's right. Here is some DBSNP databases like that. So feel free to use interchangeably if you are studying human. Okay. All right. Now, let me introduce the bed format. Sometimes we wanna say that promoter region starts from here and ending there. And what is the best format to use that? And the short answer is a bed format. But if you ask, is a bed format the only way to denote the interval? The answer is no. There are many other compatible formats, but bed format is one of the simplest and then most efficient and widely used format to describe an interval. So the example for the short bed format is, chromosome 1, starting position of the interval, and then ending position of the interval. And here is very important thing. The separation between column. It's better to be a tab key, not the single space. So if I have to make this format, let me show you how I actually can make it. Since I'm using the Mac, let me use the text edit. So I want to separate by the tab key. I want to separate by the tab key. Chromosome, separate by tab key. Starting position of the interval, separate by tab key. And then the ending position of the interval. And if you want to have a second interval, you can actually do that. And then you just save as test1.bed. I want to use rich text. It's better to be the plain text, but because rich text has an extension RTF, but if you use a simple text, you can make it ending with a test1.bed. So this is one, but if I have to do it, I usually use a command line. So you can use a Nano or BI in the Mac or the Windows, Linux. Test to the bed file, and then paste it, and making sure that this is separated by tab key. It is separated by tab key. And then this is a Control X to exit. Save modified buffer. I will answer Y for yes. And then file name is a test.bat. Yeah, that's right. So I just saved it. If I want to show the content, cat test to the bat, it is actually showing the a valid bat file. You can actually upload this bat file in the variation viewer or genome data viewer, track and user data like that. Upload file. Chromosome 1 disposition. Yep. If I click it, I come to chromosome 1 starting position of this and ending position of that. And then like that. If I want to go to this the other position, yeah, starting position is this and ending position is this. So oh, let me repeat. When I use the text edit, I didn't like that it has a RTF extension in it. So I decided to use the Nano in the text terminal. So I used the Nano test to the bed file. What is a Nano? It's a text editor in command line. And then just do it and then paste it and then Control X to save it. So if I do the cat command, and it shows the content of it. So basically, instead of using Microsoft Word or text edit or Notepad, WordPad, I use a command line to edit the file. And after doing that, I upload it to this genome data viewer by clicking this the track and user data and upload button. And then if I want to go to certain interval that I put, I can actually copy do this like that. And how many of you actually can follow what I was doing in the real time? Now, why don't I actually give the about 5 to 10 minutes so that you can actually do that? Can you actually make a bed file of this and upload to genome data viewer and then click to go there? For the this position, why don't I actually copy and paste? Let me do this. Give it to you by chat. And why don't I actually also give the test to that bed file here too? So let me just repeat. Can you make a bed file and then upload to NCBI's data viewer and click to go there? That's the class exercise. Yeah, let me show the command. Nano test something.bat file.
After you're done, can you like
And then copy and paste.
Thank you.
And Control X to exit. Control X to exit and press Y to save it and press Enter key to approve the file name. If you save as RTF, rich text format, it doesn't work. But if you save as a plain text, it should work. Yes. Yeah, let me show you that. Control X to exit, save, modify, yes. File name to write test2.bat, is that correct? Press Enter key to approve it. Yeah. Cat to check the content. Can you type the PWD? PWD, present working directory. Yeah, that's where that file is located. So can you upload, go here. Options, click options, and there is an upload file.
I'm able to work in term. Does he want something like this pretty much? Or
Sorry?
Do you want something like this pretty much? It's like,
Yeah, that's it.
okay.
Right, question?
The file was created, I uploaded it, but it's showing something different. I mean, the location is fine. I just show you my screen on the
Yeah, let's see your screen.
So for the first one, you see that it's like the location is there, but it's showing more than a different range. I mean,
Yeah, why there is the K?
yeah.
Can you show the content of your actually yeah, it shows the right position there. The pop-up like
Oh, okay.
yeah, somehow it doesn't show yeah.
Okay, so all I did was I just copied and pasted the PowerPoint into like a text editor.
So you need to have a tab space in between, not normal space. So you use a tab to get the space.
Yeah, I think
Completely cover the space first and then go
Oh, okay.
file itself seems fine.
And you see how it's looking in the
Just separate by the tab.
And once I do that, I just hit file save and I just called it test2
Yeah.
dot bed, and then
yeah.
and then when I went to my desktop and I just did AI test2.
Yeah, I think it's fine.
Oh,
Right.
I guess R.
Is there anyone who has a difficulty of what I
So then that's all I do.
Yeah.
And I just upload it online.
Yes.
So basically, what I did was I created a very simple
first do
text
SQL.
file to show the interval and upload to the NCBI's Genome Data Viewer and then show the interval.
And I just
Do a cat.
Now I have one question for
Test.
you.
oh,
You can do tab to automatically get the file name.
okay.
Yeah, seems fine.
And I just upload it online.
Yes.
Okay, thank you. That's all we to do. Wait, why is my screen sharing?
Because you're working.
I would like to show the position of a thing. So, for example, I want to make a chromosome 3 at 7 years position. I have a thing. I want to denote that. How can you prepare that file?
how long is this one?
It is one base pair replacement. So chromosome 3 starts at the first position, second position, right? At the 70th position, let's say reference sequence is A, and then in my sequence it's T. I just made it up. Then I would like to actually visualize that into my genome data viewer. So if I create that file for that purpose, what is the best way? So I know that chromosome 3, and then I'm going to separate by tab key. And then what is the starting position and what is the ending position of that info to denote the position 70. And there are actually two possibilities. So I can say 69 to 70 or 70 to 70. Do you understand? But this seems to be mathematically correct. Starting position of the SNP is 70. And then ending position is 70. That's the definition of a sequence. There's another possibility. All right. Let's do a quick poll. How many of you think the best way to represent this is number 1? Please raise your hand. 1. How about 2? Okay, 1/3 of the people. How about 3? 1/3 of the people. And for 3, can you tell me the reason why? Mathematically, yeah, this is an all 3 possible combination. And just to let you know, in this genomics field, unfortunately, we don't use 3. We use either 1 or 2. And it has an even name. This is called 0 band. And this one has 1 band. So I would like to let's do the example. Need chromosome 5 and first base pair. I would like to know that. Then the choice is these are 5 separate by half, and then either 0 and 1 or 1. This is a starting position and ending position, starting position of the ending position. And then this is replacement. The ending position cannot be 2. Ending position should be 1. Do you agree with that? So unfortunately, this is not actually correct. But these two, this is obviously for the SNP. The first base pair position is the position of the start of the SNP, single nucleotide polymorphism, base pair chain, and the ending position is this. But this is even the mathematically this is correct. This is 1 band. If you want to show that in the interval here, why don't I actually do the demonstration here? Let me make a nano test of 1 bed.txt bed file. Chromosome 5, tab key 1, 2, 1. Oh, sorry. There's screen. So I want to use the nano command again. Text editor. Test about 1 bed file that I just showed the 1 bed. SNP to show the location of a SNP at the first base pair position of a chromosome 5. CHR5, tab key. SNP's starting position is 1. SNP's ending position is 1, right? Let me do that. Control X, and then the Y, and then press the Enter key to approve it. And I'm going to upload this file to this genome data viewer. Click Options in the Track and User Data section, and upload. files. And choose file. And this one, click open. Do you see that fail to add the tracks? This error message here. Oh, yeah, fail to add the tracks. So even though this one is what we call the one-bed file, it failed to be added on there. Why? Starting position is 1, ending position is 1. How can you denote the interval? It's a thin line, and it shouldn't be visible. Therefore, people invented a zero band. And then we just subtract one in that beginning. I know that this is also possibility, but somehow people did that way. So this is in the case of a snip, you just subtract one from the ending position. So that's what we call zero band. Why does it call zero? Because if you check out the snip at the first page, it's not zero. So how do I actually make it and upload it? Yeah, that's right. Test of a zero bed. Chromosome 5, 0, and 1. Control X and save Y and press enter key. And then if I upload it again, try option, upload files, choose file, and then zero bed. Open. And it's successfully added. And now I have a snip here. Do you see this interval here? The first base pair position of the chromosome 5, 0 to 1. It has a now one, like a pixel size, one basic size of interval. So that's basically zero bed versus one bed. Yeah, question?
You mentioned that you show the specific base you change A to T. Is there a way to show that?
Oh, not here. It's a bed file just shows the interval. So it doesn't show that. But if you look at this, let me go to the test 2. If I just magnify all the way, now it just shows a forward strand and then backward strand. So in this, for example, 23401.360 position is reference sequence is T in the forward direction, and then backward direction is complementary, so A. So people usually denote on the forward direction. So we know that it's a reference sequence is T, and then in the case of a SNP, T to some other allele. Bed file doesn't have okay, that's actually a good question. I would like to specify either forward strand or backward strand. Then you have to extend the bed format. I just showed the simplest bed format, but there is 2 other versions of a bed format. So it's called a short bed format, and I show the that, and then even I mentioned the 0 based and 1 based. $49.75. 49 to 75, it's an interval, 25 base pair long interval, not the SNP. SNP should be the oh, I want to denote this interval. If you show the take a -1 here, it's you have to say that this is a zero based. Yeah, that's what starting from. So just looking at, you don't know what that is, zero based or one based, but one based chromosome 250 to 75 is equivalent chromosome 249 to 75 in zero based. So these are the if zero based of that is equivalent to the one base of the second one. That's what I just wanted to show you an example. Yeah, question?
Does the different browser databases use different zero-based or one-based?
One thing that I can tell you is the UCSC Genome browser use a zero-based, and also the this NCBI browser use a zero-based. But the other browser, yeah, I'm not sure. It depends on the author. Yeah. Other question? I heard the. So I was about to explain the plus strand and minus strand. When you use a short bed file format, you cannot denote that whether that interval is in the plus strand or minus strand. For that case, we use a long bed format, which has more columns. So in addition to chromosome start and end position, you can give a name of the interval at the fourth column and some score if there is, and then strand information, plus or minus, like that. And even there is a 12 column the bed format. Dig start, dig end, RGB code, block count, block size, block start, something like that. So you can, why does it need it? You know, there is a thick block and little thin, like a line thing, right? By having this kind of a 12-bed format, you can actually completely visualize this kind of a diagram. So to visualize this kind of diagram, you need 12 bed 12 format, long bed format. How do you get the score? What is the score? Whatever score you want to put. Confidence score, p-value score, p-value multiplied by log, whatever you can think of. So if you have to use the score in the bed file, use the bed 12 format and put the score in that column. 1, 2, 3, 4, fifth column. If you need it, just put the 0 or dot. Yep. Any question? Yeah, I think this might be somewhat complicated in the beginning. You don't have to memorize everything, but I want you to at least memorize, understand, okay, bed format is used for showing the interval in the genome browser, and then chromosome start position and ending position. And to show the SNP properly, genome browsers prefer to have the zero bed, basically subtract 1 from the start position, so that interval is not the line, but it can have some it spans, so it can actually show something in the genome browser. For example, this promoter, right? you can give any name for that interval. That's what it is. And this person wanna give a promoter for this interval. Starting position of a promoter and ending position of a promoter. But you can give whatever name. You have a freedom to do. Okay. So yeah, I'm not that much worried about the BED12 format, and BED6 is not needed. When I actually grade it, I usually grade based whether you made the first 3 columns correctly. But if you need to specify more information, you may want to use the BED6 column, BED12 column. Yep. Any question? No. So now I'm left with 2 things: weak file and then the explanation about the group project 1. And do you guys want to take a short break, or do you want to continue? Sure. Okay, let's do the quick poll. How many of you want to take a short break, 5-minute break? Okay, 3 people. How many of you? 4 people. How many of you want to just move on? All right. Yeah, let's just move on. I think what you want to do is let's try to finish and get it done and go home early, right? Right. So, right. Now, I show the bed file for the interval, but there is another format, weak file format. And I explained the FASTA, right? FASTA format to show the sequence, starting with the greater than sign, ID, and the ACGT sequence. And I explained that bad format, now weak format. And when you reach this, one of the commonly asked questions is, how many formats do we have to memorize? Maybe about 5 more over in this semester. Yeah, it's like the getting to know the format is important. Everybody knows PPTX is a PowerPoint. DOC is a MS Word, right? So do you always say, hey, give me your presentation made by Microsoft PowerPoint? Or do you just simply say, can I get a PPT? You actually ask, you actually communicate with a DC extension, right? Or can I get a CSV file? Do you know what the CSV? Comma separate value. So you guys are always already using this extension, right? When you do the bioinformatics, most bioinformaticians wouldn't say, can I get the interval written in some format? We just say, can I get a bed file? Can I get the FASTA or FASTA file? That's customary to say. So, so it's, even though it's kind of annoying, you actually have to know this format. So that's why I'm introducing this in like a bit more detail. Bed file is much more important than weak file, but still, weak file is often used in the chipset, promoting in the specification, followed by the DNA sequencing. So that's why I want to introduce this chips, no, the bed file, if I do the genome browser, you can make this kind of info from this starting position and ending position. If I ask about the height of that, there is no height basically. It's either 0 for non-interval region or 1. You understand what I mean? But sometimes when you have a biological data, sometimes don't you want to draw something like this kind of, this x-axis is the base pair position. First position, second position, third position, like that. Sometimes, don't you want to show the binding, like the, bind affinity or those kind of things. And if you do that from the data, does it look like that? Not like this. So sometimes when you do the bioinformatics, you might actually want to draw something like this. But that file is not appropriate for that. Then wiggle file, big file is appropriate. It's very simple. Position and height. Position and height. If you look at that. 3796844th position, the height is 0.6. And the next base pair, height is 0.8. Next base pair, height is 0.9. And if you just save it and just call it wig file. And if you visualize in the genome browser, upload it, and then like that, it will show the wiggle shape. What do you mean by natural?
Like, naturally, the way that it's doing it. If you just want to visualize it.
Visualize height.
Yeah, fine.
Yeah. So 0.7, 0.6, 0.8, 0.9, those are height of the figure. So 1.2 or 10,000 can be anything. Usually we get that number from experiment, like a binding affinity or something like that. Pardon? Yeah, number of reads. In that case, it should be integer. Okay. So if you want to actually show that this kind of trace, and that file is not the right choice, wig file is the right choice. And I would like to mention the two more things, which is not written in the in this PowerPoint. It's the additional information. If you prepare this way, how many rows do you need to have for a whole genome? 3 billion, right? 3 billion rows. Is there any, do I really need that kind of resolution? In many cases, no. So one trick that people made is, how about every 10th position? So just keep the first position and just ignore everything, and then 11th position and then 21st position, like that. Or every 5th position, or every 100th position. So in that way, you can compress the wig file. So that's already often used. That's one way of reducing the file size, because 3 billion rows are too much. Yeah, question?
So what about the pattern between the rows? How do you miss all the patterns?
Yeah, that's a good question. If you use the every 10s or 100s, a lot of things are being empty, right? So if you want to have every base pair resolution, this kind of thing is not appropriate. So you have to extrapolate. So in that case, even though you don't want, you have to use one base pair resolution. No. Okay. Yeah. But Is there any good way? I don't want to have that every tense because of that problem. Do you guys use a zip file, zip? When you compress it, if the file size reduces, right? Why don't we actually pick the one base pair resolution and instead compress it? And did you have to know that there's many compression algorithms? Which compression algorithm would you want to use? And have you ever heard the gzip? Let me show that again. Have you ever heard of gzip? This is probably the most popular. If you compare FASTA file, ABD.SA, and if you compare with the gzip, we usually give a stable number. It's the most popular use. Popular use the compression. But you know that most popular doesn't mean that most efficient. Right? So somebody uses the other the more efficient algorithm, but this is in genomics most people use it, even though it's not most popular. Pardon? Anything. So for example, ABCD fast will not compress with the G. And let me show you how to compress it. It's pretty easy in the command line. If you can compress anything. Gzip, test1.bat, ls minus. And then it actually created this file. So if you happen to see .gz, you just simply recognize, hey, this file is compressed with a gzip utility or software. You have to install the OS-dependent gzip software. But for the Linux or Windows, for the Linux or the Mac, gzip is installed by default in most of the case. So since I'm using the Mac, I can just use that. For Windows, you might need to install the WinZip or those kind of things, 7-zip, bzip. Why do I just mention that the zip compression algorithm? One way to compress is like that, but that does not have a one base pair resolution. Instead, you can use a regular big read file or bat file, and then compress with the gzip. That's one way. But another way is you use a somewhat optimized for the WIG file. That's called a big WIG. In short, we usually use PW. So that file has its dedicated compression algorithm, and then it's called a big map. Or we often say .bb. I just introduced this, but the reason why I didn't put it into the PowerPoint slide is first of all, at this time, I don't think that you really need to know, memorize your own all the time. But I just want you to be aware. Yeah, I heard about that Sijung was talking about that. But when you actually see, oh, yeah, I know what is the bad file, but I don't know what is the big bad file. But you simply say, that is a complex version of the bad file. What is the big weak file? That is a complex version of a weak file. Because weak file tends to be very large weak file. But, oh, I didn't erase this, sorry.
That's what you example.
Oh, that's right. Example of weak format. Yeah. Span is the interval. So every one base pair resolution. If it's the span is 10, it's every 10 base pair position. This value actually not be regular, but variable. Like a 44, 45, 46, 47, that's not variable. But by having this, you can have some variable step. 44, 45, then not the 46, but you can skip the 46 and yeah, that's right. That's right. Yeah. Right. Now, do you have any questions so far? So basically, that is the content of Unit 3. If you do not have any question about the Unit 3, I will go over the assignment for the group project, which is due on coming Monday night as the group.
This one?
18th, sorry. Yeah. So this by this Saturday, I think you guys already formed the group. So I'm going to finalize that is your group formation. That's good that that's one of the good things that we are in the in-person. Because in the online setting, this is not easy. Yeah.
Also, when you do the paper analysis, like didn't update on the calendar. It still says like the 11th.
Oh, really?
Yeah.
Yeah, but yeah. Yes, Sijung is usually checking the making the calendar, so I don't have access. But why don't you just use this? Yeah, the syllabus is the one that everything is based on. Yeah. So this is a syllabus, a group project session. Yeah. Yeah. Now, this week's the due by 18th of September is your group read one paper, a benchmarking paper, PMID of this and this or this. You just choose one and then write the brief summary of the paper. So why don't I actually show just a middle example? PubMed.gov and just copy and paste that PMID. And this is the SNP. A novel SNP, like a paper related to the Alzheimer's. So you just read this paper and then make a summary of this. And starting from this year, I have to worry the ChatGPT, like a prompt, hey, summarize this paper. Yeah, and yeah, I don't know how to distinguish your work versus AI work. But you have if you, I'm not prohibiting using the ChatGPT. All I ask is if you use ChatGPT, cite it properly. Double quote for direct quotation, and then if you paraphrase it, yeah, you can just follow the APA style. So let me move on to the here the group page, group project session. Yeah, and this is actually not me. Actually, he's Sijung. Just in case you might wonder. Here is a group project. So here is a specific example. I put some more the like a PMID here, but if you prefer some other paper, just let me know. That I usually honor them. The purpose is very simple. Why don't we actually start from the example? We are studying a novel SNP like a case, right? So there should be some papers written researching on the novel SNP. So here are some examples. Why don't you actually read about it and then discuss and then summarize and submit that? And I just want to explain the extra credit assignment. So you will prepare one master PDF, which contains the title of the paper and then the summary of that thing. So that is like a PDF, just submit it. But in addition to that, if there is optional extra credit assignment, which is making a team charter. This is what I mentioned. When you actually run a group, I want the everybody efficiently and happily communicate each other. So it doesn't matter which method you use, text, Discord, the MS Teams, or phone call, or email, whatever. But I want you guys actually agree on your preferred method of communication, and then define. So that is how to communicate. And then if you actually want to define the role, hey, I'm going to be a team leader, or everybody participate equally, not like a team leader versus actual team member structure. Everything is you guys can form yourself. And then I want you guys discuss about what will be the role of team members, right? That's what I mentioned. And also, if there is any conflict, what is your how would you guys resolve that? If you think about that before and then write about it, and that might reduce the chance of having trouble later. And then especially this group project has a deadline due date, like a Monday night. And then if your group do not communicate nothing at all until the Sunday night, and then everybody starts on Monday, that's not really desirable, right? So what is your group's policy or their way of doing regarding that? Since due date is always on the Monday night, so by Wednesday we do the preliminary study or something, so you can actually think about it. So I'm not going to make I'm not going to order anything. You guys like decide yourself. And here are some other like the yeah, references. Yeah, question?
More details should be some
and that's not written here, right? That means it's on your discretion. But the more detailed, it's better for you later. Because if you just prepare one paragraph summary, you can probably do it in like 5 minutes. Yeah, but why do we do this? Because you are going to excel. You wanna apply for your own SNP, and this is a benchmark paper, and you wanna actually do better than the benchmark paper in some aspects. So if you know how the previous authors did it, example, if you analyze very well, then we give a very good guideline on how you can actually come up with the future. So when I actually grade, I try to give as much as credit possible. But the most great point is actually at the end, the final draft. So you probably get the most of a full credit for this small assignment. But that doesn't mean, even though there is no requirement, okay, I just wanted to get it done in 5 minutes and I just want to write the 5 sentences and that's it. Even though you can technically do that, but that's not on your own benefit. Thinking about this whole semester long project. So, but I will let you guys decide. Not because of the grade, but because of the overall like a fulfillment and then successful like a completion of the project. Okay, yeah.
Around that means there's no limit. I could write as much as I want, or as I want.
Yeah.
Okay.
Yeah. Yeah, there is no upper limit, either there is no lower limit. And then I will do the minimum involvement for your grouping. When your group really goes wrong, then I will involve it. But but I try to you guys do the group work yourself. Yeah. Yeah, that's basically all the content. Do you have any question before we close? Right. Yeah. See you next week. Yep. Thank you.